All right, welcome back. I hope you had a nice break with these fantastic cinnamon rolls. There doesn't seem to be enough. So Jessica Storer is going to introduce you to Dfam.
Thank you, Clement. So you do not need the virtual machine for this particular presentation. I actually switched the virtual machine portion of Dfam to repeat masker, so it's a little bit, just because the Dfam one was a little bit long, so I had to switch it over. There is some overlap. So because I don't trust that the virtual machines or internet's going to work perfectly every single time, I'm doing a kind of a split screen. I will make this publicly available, the presentation that I have. I will make it publicly available, so anybody that wants this particular presentation, instead of just taking their own notes, totally fine. This is all published, it's just my notes on top of it. So if you could please, if you want to follow along, interact with the website yourself, please make sure, one, that you have an internet connection. If you don't have an internet connection, buy one. Please talk to Clement or Alex or Travis, somebody that can actually help you for the technical part. And please go to the Dfam website, https://www.dfam.org. We're going to be doing, again, kind of parallel for both. So to kind of introduce myself, I'm Jessica Storer. I work with Arjen Smit at the Institute for Systems Biology, and I am a Dfam curator. Currently, I handle all of the community submissions. I love what I do. I love working with science. I truly do. I love working with different scientists. I love working with all the different organisms. One of my favorite people is, shameless plug for my favorite person, Clement Goubert. He was amazing to work with the rice weevil genome and the TEs for that. And it's like everyone should be like Clement for like submitting TEs. He's giving me a no. Okay, so I want to make sure that you guys can get the most out of Dfam, and it's a very rich resource. And to know all the bits and pieces of Dfam, more so than just searching, or maybe there's search parameters you're not aware of, or how to access some of the data. Okay, so I already have Dfam up. And so we're going to go, so there's three parts: website basics, submitting data, like getting an account, and then interacting with the data and downloading things via the API. So that's if you have an internet connection. Then you can download things directly from Dfam. Not download, but interact directly with Dfam. Okay, so we're going to go straight from left to right based on the arrows that you see above. Okay, so the first is just, oops. The first is just the home page. Oops. Oh, well, sorry, we'll get there in a second. But what makes Dfam different, why is it this particularly special database, and I think it is, I also work there, I'm a little biased, is that TE families are defined by seed alignments containing prototypical copies. So you maintain the consensus or a pseudomolecule in profile hidden Markov models. So this is important because there's this transparency of data. You can see exactly which instances or copies in the genome are contributing to making the consensus. Okay. Also, we've shown with a couple of the different publications, 2013, 2016, or 17, depending on when it actually is available online, and then a 2020 publication shown that this has enhanced sensitivity with profile hidden Markov models for the in terms of finding copies in the genome. Okay. Also, lots of diversity in Dfam, and we want to keep growing, especially with the curated data sets, as Arjen kind of highlighted. The uncurated or raw set, which is just a repeat modeler run, and it's submitted to Dfam, we extend a little bit, and that's it, versus curated. We'd love to have more curated. And the reason I say we'd love to have more curated, I think is the obvious reason, is whoever's submitting it. So for example, someone was speaking to me earlier about the axolotl genome and the TE models with that. We'd love to have that highly curated. It's nice to have that expertise involved with that curation level and just some of the raw data sets. And that's part of what I like to do, is interact with the people that's their genome of expertise. So like, you know, tell us what the TE should look like. We would like your knowledge. So far in Dfam the version 3.6 in April 2020, we have several 100,000 models. Only a fraction of those are curated. Robert Hubley, who is not here, was telling me before I left that DFAM 3.7, set to release in the next couple of months, is going to increase our species, double the number of species we have available, and then increase the number of models available by 3.3 times. So we're excited to continue to expand this resource. I'm working with 600 plus insect genomes. Alexandra Pascal, he's going to help us increase our plant genomes by 100. Very excited to continue to expand this. Oops, no, wrong one again. My goodness. Please go to the next one. Okay, so in terms of the website basics, hopefully you've already found yourself here in the DFAM home. Just to kind of explain a little bit of the structure, if you hit the DFAM logo, no matter what page you're on, you'll go to this homepage. Okay, there's some quick links. Mostly people would like to either search or browse or find a specific DFAM entry or like a keyword search for an entry. Okay, so some of the broad quick links. Anything that I'm talking about today or specific information that you don't think is included in here or something, some niche question is on the help page. Okay, so it's going to be, well, you just have to click on help and there's different categories of help. A lot of detailed information in there on top of if you would like to communicate with me directly. Okay. Also, we're going to talk about this in the second portion of this short demo, talking about logging in. I have the workbench open on because I've already logged in, so you guys don't have to watch me fumble with my password. But that's what it looks like when you're logged in. You see a workbench. But if you're not logged in or you don't have an account, I'll kind of show how to do that, but it should say log in instead. Okay, so the first aspect is searching for a sequence. So for this specific example, I've just clicked example. Oops, I guess I could do there too. I've just clicked example, so you can just click an example. I have chosen the DFAM curated threshold. For more information on thresholding, I have a couple slides at the very end if you're interested in learning more about what the gathering threshold or trusted cutoff would be. If we don't have enough time, I'm happy to talk about that later. And like I said, I will make these slides available, so you'll have that information at the end. Okay. So the default is the search box is empty. I've clicked the example. I'm just going to go with the DFAM curated threshold for the purposes of this demo, and then hitting submit. So it's probably going to take a little bit of time. I also was anticipating that maybe this isn't the only search, because if people are all searching at the same time, yeah, okay. Just going to make this a little bit bigger to get around the wait time. Okay, so what should pop up is you're going to, oh, it did actually pop up, great. So what you're going to actually see, you're going to see the particular genome. In this case, it was Homo sapiens. For the search, it was a trusted cutoff. It's different if you have a specific e-value, NCBI kind of e-value, what you're expecting, when the job was submitted. And so what you're going to see, actually here it is on the website as well, what you're going to see is you have some colorized bars up above. If you hover over, you'll see that specific match for your sequence. In this case, it is L1ME3G, 3' end. I'll explain what that is in a minute. So this is the positive strand, which is above the gray bar. Negative strand is below, and tandem repeats are on the gray bar in black. So tandem repeats show up at the very bottom of the page, if that's what you're interested in. Okay, so if you were to go and click down for an example, what you're going to see is the model, which is the DFAM entry that matches this particular portion of the sequence in which you're interested in. And then the example sequence is the query. Okay, so the query is your sequence that you inputted. In this case, it's the example sequence, and it was human. Okay, you're also going to see the posterior probability, and there's again, I don't have enough time to go in all of the detail, but there's a specific stars have the highest score, and then there's a numeric score. You guys, wherever work with SAM or BAM files, it's kind of the same scoring system. Which, again, if you don't know what that is either, I'm happy to talk about that later. You can also go ahead, you can also download this data. It will download all of it. So not just one entry, it will download all of it. and it will be in the same format, like this table format unclicked. And we'll also show the DF number for that particular entry. I say DF, that's for this case, it's the curated entries. So you also might notice that instead of saying it's just an L1, it's the 3' end. So I will get into much more detail. Again, these are linked, and it's also a shameless plug to come look for just to come watch the repeat masker one. But the 3' end, because of the biological nature, because of the biology and the movement, the target prime reverse transcription mechanism for lines, you're going to have 5' truncation. So the most reliable way to identify L1s is to kind of, and the way that repeat masker stitches things together is to have the 5' end, the 3' end, and the internal, and the linker of like the ORF2. So, but again, questions after and or later coffee breaks. Happy to talk. Okay, so for the next, oops, please work. Okay, so next is going through is still, so this was sequence search, and then there's the annotation search. So for annotations, you're going to have a specific assembly. We do not have, for the DF records, the curated records, we have these assemblies. So you'll be able to search those. So for example, for human, you can just put in 1, 2, 3, all the way up to the max chromosome for human. So I'm using, again, thank you, Kamant, for rice weevil for the example. If you want to figure out, if you want to isolate and retrieve annotations for a specific range on a contig, you have to use the GenBank GenBank contig names. So for example, this might correspond with a specific chromosome in rice weevil. We don't have the number like 1, 2, 3, 4. We have this NW underscore number. Okay, so that's what we have in Dfam. So again, if you want to use this on your own, search your favorite genome. Make sure that you're relating them back to the GenBank coordinates. Okay. Again, still available for view. So if you were to search this, it's the same viewing for the sequence search. So same output that you would see, but just for that particular range. Okay. All right. Okay. So this is so no notes on it is the browse page. Okay. So kind of going over each of these pieces. So if you're going to search for a name or a session number, you can search for the DF record or the name, such as MIR, ALU-Y, ALU-SC. For the classification, it's going to be the Dfam classification, and there is a translation table for that if you're not familiar with what that looks like. So I could, for example, I could type ALU. It's going to pop up with something that will match the classification scheme, and I can click and it'll start searching things. So if you notice, and we're going to go through the API later, but there are 190 results. And if I hit ancestors, I think it's the same, including uncurated, the number goes up. Okay, so make sure that you're trying to search the appropriate taxa, in terms of like ancestors and descendants, and if you want to have uncurated data as well. So for example, if you're searching Homo sapiens and you don't have a classification, you just want to search for humans. If you just look for descendants, which is the default, you'll have 29 entries. If you want ancestors and descendants, you're going to get 1,356 entries. So make sure that you have the kind of understanding of what that's going to look like in terms of the results. Okay. So also, because there's a lot of information contained in this database, if you try to search for a taxon, make sure that you know that there's going to be a bit of a lag. Okay, so just like let it fill a little bit. Also, my computer has a tendency to autocorrect. So make sure that the autocorrect doesn't try and put in home instead of homo for Homo sapiens. Mine does that. It's very unfortunate. Like, wait, there should be human entries in here. So make sure you're double checking that or turn it off for your searches. All right. So we're going to go through a specific example. So if you do uncurated, obviously you're going to get the full database. We're going to go over a specific example of harlequin found in primates. Again, you can also, not again, but you can organize the results based on sequence length. For example, if you have an idea of what the full length sequence for your particular TE class is, you can search those. Okay, limit to a certain length. Okay. So if we were going to start this over, please and thank you. No? Here we go. So we're going to go to Harlequin. I just click on the DF number, and this is the summary page. So I've have the window a little bit shorter at this point. And so anything, if you want to kind of go to different portions, there's a drop-down menu. Okay. So in terms of the summary, just a brief description, not to be confused with this particular description at the top. These are some of the curator notes. I believe these are actually Arian's notes. For Harlequin, no? Could be. It has his cadence of writing. I could be wrong. I could be wrong. So this will come up later for the API for classification searches. This is in the entire path for the classification that's listed here. And if you click on this green portion, also seen here, it will link to the classification page and have that all the way expanded for that tree. You can also have an external link to there's an external link to the NCBI taxonomy, so you can see the entire path it would take to get to primates for this entry. You also have an alias. So let's say, for example, the rep-based name is slightly different, and it might be a little bit confusing if you're trying to search for a specific subfamily. We have aliases listed as well. All right, so going through the seed, okay, so we have the seed alignment, you have the coverage at the very top, you have the instances, so it's one sequence per line here, and the scoring is the alignment quality over a 10 base pair non-overlapping window. Blue is the highest quality, red is typically actually a gap, but more low quality alignment. Okay, so we're talking about the seed alignment, representative sequences. This is what we mean, or this is what I have been meaning this entire time. Okay. So then if you have a coding model, going to features, there's two types of protein alignments that are available. There's the curated, so this doesn't have a curated protein sequence, but it is curated and then mapped back to the sequence. Versus the aligned, which we have the TE protein database that's aligned to the sequence. Okay, it's not like mapped back, it is aligned. Okay. Note, there's only a portion shown here, but generally a feature is a protein coding region. We hope to, in the future, to include target site duplications and other structural features that kind of, are truly features of an element. Right now, it's just coding. So you wouldn't see this for like an ALU or any type of sign. I keep bringing up ALU because that was a big part of my dissertation work. So, all right. So the other thing is you can actually so the consensus is available, and you can also see see the logo, so the conservation of the consensus sequence across all the different bases. But one of my favorite portions of things is actually searching for a specific range or motif. And so you also, this doesn't occur for all models, but specifically for Harlequin, you have this model mask. So basically what Dfam is, Dfam, the inner workings of programs, it's identified as a false positive attractor. I'll show you why, but alignments are not seeded in this region. So if we go ahead and put in that specific range in the consensus box, it highlights in green where you have this 889 to 1080 highlighted in green. I'm not sure if you can tell, it might be a little small. It's a simple repeat region, which means it's a false positive attractor. It's just a little hard to see, but essentially it's just AG with different pattern. Okay, so false positive attractor. I know it is a little bit small. Hopefully you may have your own screen, search it up here, search it on the screen, on the website yourself. Will probably be helpful. So if we go down a little bit, so here we have some of the thresholds I've been alluding to. And again, probably won't have time to get through all of the slides today and everything I would like to get to, but they're supplemental at the end. So what you can see is the entire alignment here. You have the non-redundant coverage. So there's competition between models. You see this non-redundant coverage based on the two thresholds, gathering threshold and the trusted cutoff. Again, there are one at the end of this presentation, but two highlighted in the first paper, 2013, goes into a lot of detail, exactly how those are calculated, if the slides at the bottom are not sufficient for you, which is entirely possible. But what you can see is this portion that was this false positive attractor has this lovely spike, which kind of and indeed there's a false coverage spike here. So false positive attractor makes sense that you don't want to seed in that particular region. Okay. So here's some of the statistics. Going to the next portion for annotations. So you have this like karyotype for each of the for annotations. And it will also because it's found in primates, it's going to search all of the primate genomes that we have available for annotations. So you might not be super familiar with what each of these are. Homo sapiens, hopefully you know that's humans. Microcebus marinus, that's a gray mouse lemur. Papio nubis, baboon, Iotis nancy-mai. I can go on and on. I'm very familiar with primates, not going to. But what you can do is you have two different types of hits. You can, so this is Homo sapiens. You can have non-redundant hits or all hits. Okay, I'm a bigger fan of non-redundant hits. But the other thing to note about annotations, and just to say that we can always improve everything, or you can see the differences between different groups. So I've highlighted some New World monkeys in green, and the owl monkey, Iotis nancy-mai, Calothrix jaxus, marmoset. You can see the divergence here is a bit higher than you would see in human. So maybe we need to polish the model, polish the taxonomy a little bit. You know, still things to be learned. If you go even farther away from human and you have lemur, I won't go over the names again, but these three lemurs have a much higher divergence than even the New World monkeys, which are a bit closer humans. So just we can actually use this. There's again rich resource. We can use this information to kind of polish the data, continuing using this information. Okay. All right. So then there's a relationship tab. So within the relationships, you can actually, I'm not going to include uncurated. It will probably not load super well. But what you can do is you can organize by %ID or coverage or e-value. So I think right now it says, yes, it's all Dfam. You can click only related clades. Okay. Looks at the same at the top because of the ID and the coverage, and the % coverage are pretty high. But I wanted to, so one of the other features is you can actually hover over and to show exactly how these sequences are aligned to each other over what region. And so I've selected this region particularly because of that false attractor region. This is exactly where it aligns, and it's that just to show that, oh, it's similar here. So it's likely shared between subfamilies or related families to Harlequin. That also is a false positive attractor for simple repeats. So, oh, there. And you can hover over, and you can also, if you would like, you can actually click on this family, and it goes to a separate link. If you would like to see exactly what that particular related family might look like. Okay, now downloading the information. Of course, I went a little too far. So downloading information, there's many different data types available. If you just want to look at the FASTA consensus sequences, that highlighted in blue. Oh, I didn't do this quite perfect. So you have the FASTA sequences, which is the caret title sequence, like our favorite format. You also have Stockholm, and Stockholm is what is actually boxed in blue below. You have a lot of metadata information, including classification, author notes, publications associated with it. For this particular one, it's Waterflea. It happened to be a raw data set that I chose here. You can also have the EMBL format, which is accepted by Repeat Masker in terms of a library. And the EMBL format is boxed in red here. And you can download redundant or non-redundant data sets for all of the associated genome annotations. Okay, Classification, really cool portion. Okay, so there's 2 classification categories under classification. If that made a little sense, if that made sense, what I just said, apologies. So you can hover and click. So you have accidental, you have interspersed repeats. You can go to pseudogene, transposable element, unknown, and you can keep clicking out. Or what you can do, if I wanted to search alu, again, my favorite element, it would expand all the way out to show exactly where this is, alu. Okay. Again, it's embarrassing how long I went without knowing that. It wasn't like I learned last month, but it was like a year into being with Dfam. I've been here for 3. So to be fair, it's not just I've been here a year. So that's pretty cool. You can interact with the tree just to kind of figure out what the classifications are, or you can put in a specific element. Again, there's also a translation table available to kind of, Piegu arenas. No, not arena. I'm sorry. Arena's still awesome. Wrong person. Wicker, Crucio, and then, this, but then there's kind of, there's not always a one-to-one for everything. This is trying to kind of combine those. Okay. The other is the DNA transposon termini, which are hand curated by Aryan. So he should deserve a round of applause for a lot of this data. Not right now, but thank you. You're gonna start like not right now. Wait until the end. But the idea is that if you have a non-coding transposable element, specifically DNA transposon, DNA elements, you don't have a coding region. They actually do have they can have a nice conserved termini. You can download this data rather by clicking this lovely green link. Ooh, apologies. And if you were to scroll down a little bit, click on one of these models, you could see exactly how conserved this is. I've used this many times trying to find the exact edge of a DNA transposable element. Sometimes I'll sequence gaze, sometimes I'll use this as an actual sequence search, cross-match, blast. Very useful. So let's say that you don't want to browse, you just want to download everything en masse. For this specific release, you can go to the download page, and this will pop up, right? If you want to just look at the families, we have this H5 EMBL format, curated on only if you want to have redundant or non-redundant data sets. I highly recommend downloading the H5 format. It has a lot of data associated with an NCBI taxonomy, has a consensus sequences in it, has HMM profiles. It has so much data included, rather than just having one data type. So I recommend using this particular, downloading that particular data set. There's also release notes, like what's the difference between Dfam 3.3 and 3.6? Here you go. And between all the releases, you can look at this information yourself using, again, it would be release notes for 3.6. So here. Right. So submitting data. I think I have like 5 minutes. No, I have 10 minutes. 10. Yeah, okay, sweet. So if you want to log in, so submitting data to Dfam can occur in a couple of different ways. So if something's going wrong with your data submission, you can email us directly. Preferred way is to log in and then submit data that way. So the process includes logging in, like clicking there. This is what will show up initially. If you need an account, you're going to click need an account. And what happens then is you put in your full name, email, and password. You click register. And for me, what happens for me is I have to go here. I click users, and then whoa, my goodness, woo! And then I would click approve submitter. It's a bit manual, and that's great. I hope that these are all people from this conference. Yes, this is great. This is fantastic. But that's how you would actually register for an account. Free to do so. So this would show, this is what my page looks like. And this is what the submission page looks like. So the more data, the better. Again, small text. I really apologize. If you're looking at the website, you're probably able to magnify that a bit for yourself. So in terms of uploading the data, you can submit it in either consensus sequences or Stockholm, prefer to Stockholm. We will have to regenerate the seed alignments if you can submit consensus sequences, in which case we will need a genome, preferably a public assembly, or at least tell us what you'd like to do, or in terms of timing of data releases if you have a private one. So suggested information, but please include it. Publications, very detailed information on how you generate your data set. That helps so much because 5 minutes, because I will bump the data back to you. Because I will send an email with like, I don't know what you did. I don't know why our scripts aren't working. Okay. So in terms of processing data, there's 2 different types. You've got your curated and uncurated. We have the least amount of work to do. If it's curated, seed alignments, no change. So nothing, minimal work for us to do. So Stockholm format, we talked about a little bit. This is, again, seed alignment, but like in a more HTML format up in the right-hand corner. So the Stockholm contains a lot more information. We don't have to find the instances. If we have to go back and find the instances, we use repeat masker and your submitted library consensus library to find those seed alignments. We regenerate the seed alignments. In that case, if it's uncurated, we have to extend into the flanking sequence. In order to keep it similar to what you've submitted, please submit curated seed alignments. That'd be fantastic. I'm just going to skip a little bit because I would love to get to the API. So the API, whoops, come on, Dfam. Help API. So the API is a way of, if you have internet, it's accessing the data. It's a little finicky if you have, depending on which bash shell that you have. If you want to go to the API docs, you would click this particular link. Okay, but it is just a reflection of the Dfam database. But if you don't want to interact that way, you can interact with the API. Okay, so in this particular example, you can hit right here is where families, and you're going to get families. So that's multiple in this case. If you want to do just one, if you want to look up one family, it's curl-s, single quote, API after org families, the area of Dfam you want to search, and the number. I just chose the first one. This is JSON format that shows at the beginning. Okay, that's parsable. If you want human readable format, you can actually choose a different format. I refer to the API docs because I believe I'm running out of time. But if you want to look for multiple families, you have this lovely option start. Okay, and then for this particular classification, this is what I meant by using the entire path. So for the classification, if I want to search for alus, I have to put in this entire path. If I want to search for signs, I have to put in all of this. Okay. So this would be equals, then all of this to search. The output is in JSON format. You have a total of 190, which is the exact same number if you were to browse Dfam itself. Please go to the next one. You can also, if you want to do a specific clade, or you want to have clade relatives, so I just chose mus, 20 entries if you have ancestors and descendants, descendants only 6 entries. You'll see the same JSON format. If you want to do FASTA output, again, adding and FASTA format, this is what you'll get, raw and uncurated. If you want to do just curated raw, you need another perimeter. Yay, I think I just made it. He's about to frog me. Okay, so I really hope that you feel a bit more comfortable using Dfam as a database. Couldn't get into the whole thing, but yeah.
Thank you, Jessica. Amazing.